Could we get started, please? It's on. Okay. Well, I'd like to welcome you all to the International ISB International Symposia. This year's symposia is on systems biology and the microbiome. And what we usually do is give a view of whatever the topic is, the microbiome, from the point of view of systems biology. So I'll take a few minutes and do that and thank some people, and then we'll go right into the keynote talk by Craig Venter. So I think there are enormous challenges that the microbiome presents us. And I have a list of 8 of them here, and we're not going to go into any detail now because I'll end up by discussing them in some detail. But they do have to do with new technologies that we need to develop and or the extension of old technologies. They have to do with dealing with signal-to-noise problems, which the biggest challenges are understanding the nature of the biology. They have to do with the dealing with the computational analyses you need to handle the data sets and integrate it, and of course, ultimately, you need to be able to make predictive models. And these are all things that I think systems biology is in the middle of. I would argue that the grand challenge in any kind of biology these days is complexity. And that complexity arises as a natural consequence of Darwinian evolution, a random and chaotic process. It builds complications on top of complications. There is in biology no law of Occam's razor. And of course, fundamentally what this means is biology cannot be deciphered from the top down, because no matter how smart you are, you won't know what Darwinian evolution has done to solve particular kinds of problems. And indeed, the only way to really solve complexity is enormous amounts of data from the bottom up. And one of the areas that I'm really interested in is medicine, systems medicine. And my view in 10 years that each of you as a patient will be surrounded by a virtual cloud of billions of data points, and will have the wherewithal to extract, mine, integrate, and finally create models for each individual that will optimize wellness and allow you to deal more effectively with disease. So we deal with complexity by generating lots and lots of data. But there's more than just generating lots and lots of data. And indeed, what systems biology was really founded on, at least here at ISB, are these 5 basic tenets for dealing with complexity. So one is the idea of looking at biology as an informational science, and that's really essential because that's the first reference framework for really being able to deal with complexity, and we'll talk just a little bit about that. The second idea, of course, is to create the proper infrastructure for carrying out systems biology. And I think that falls to the very heart, in essence, of what differentiates ISB from most other centers of systems biology. The third thing is understanding experimental approaches, using global systems integrative kind of techniques. And the fourth deals with the fact that technology is necessary to open up new areas of data space so we can solve new problems. And of course, we need the analytic tools to be able to solve these types of things. So let me talk just for a few moments about biology as an informational science. And we all know that there are 2 types of information. There's the digital information in your genome, and then there's the environmental information that comes from the outside. And these 2 integrate together to create your phenotype. And what connects the information with the phenotype, of course, are 2 structures. One, biological networks that capture and transmit, integrate, and modulate information. And then 2, the dynamic molecular machines that execute the functions of life in response to signals from the network. And of course, it's the dynamics of these things that are at the heart of this initial phase of systems biology. And it's these dynamics that are really driving a lot of the systems technologies that we're going to need in the future. There are many levels of information, as we all know, from DNA all the way down to ecologies. We also know the environment impinges on each level of information. And so in order to understand a system, you have to understand not only the digital core signal, but you have to understand the environmental signals throughout this information hierarchy as well. And to get them, we carry out a process called data integration. It's one of the grand challenges of systems biology. We just barely beginning to learn how to do it, but it's utterly necessary if you're to explicate the two types of information. What is obviously really important is the idea that the biological networks are not one-dimensional, but rather there are genetic networks, and there are molecular networks, and there are cellular networks, and there are networks of the organs, and finally networks even of the individual creatures. And we have to understand how the systems operate at each of those levels, how each of those levels is impacted both by digital information of the genome and the environment, and those in the end have to be integrated together to give us a total comprehensive picture of what in fact is going on in the organism. And in fact, it's these networks that allow us to organize information, to integrate information, and actually to deal with these challenging signal-to-noise kinds of problems. Now, from the point of view of how do you create an infrastructure for systems biology, I think one of the fundamental ideas is systems biology has this holy trinity that biology drives technology, drives computation, and if you're successful at doing that, you really can revolutionize biology. To get that, one, you need to create a cross-disciplinary environment with all of the scientists listed in the lower right-hand corner. And two, those scientists need to be able to speak the language of one another and as well work effectively in teams. And ISB has taken 11 years to begin learning how to do this really effectively. Number two, we have put together data generation platforms with data analysis platforms to apply to model organisms and ultimately humans. And we've succeeded, I think, very nicely in democratizing for the individual scientist ISB the data generation as well as the data analysis tools. So those technologies and their analysis can be used either for big science and or for small science. And of course, a really fundamental principle is this idea you can't attack problems in humans generally in a dynamical manner. And it's dynamics that's really the key to dealing with a lot of the signal-to-noise problems. And that's the reason why model organisms are so important. They are organisms in which you can initiate an event and follow it through to its completion, looking then at the dynamics of these so-called networks of networks. The idea of experimental systems biology, of course, has to do with three major kinds of things, as you can see. One, you need to really define the elements and components that are present in these networks and so forth. Number two, you have to actually be able to understand their dynamics, as I said before. And then number three, you really have to be able to model them graphically and or mathematically to go after and understand their emergent properties. And putting all of these things together is what I think the few centers, and I would say parenthetically, of the 70 or 80 centers that are claiming to practice systems biology in the world, I would say there are 4 or 5 that are doing it as I see it. So the essentials of systems biology then are 3. take a complicated system, you get all the facts, you create a model, and then you form hypotheses to test that model. And you iteratively add back the information you get to the model until you bring experiment and theory into apposition with one another. So I'll adjust this. Just to close that up. Okay. So can everyone hear me now? Okay. How do the systems view actually look at data features? And I think the elements we've kind of discussed. One, the analysis should be global to the greatest extent possible. Two, we have to study dynamics. Three, we have to integrate different types of data. And four, we have to deal with the noise. And I'll say there are fundamentally two types of noise. Technical noise that deals with gathering the data, but biological noise that deals with the fact any particular phenotype you assay in an organism is almost always the sum of several different biologies, and you have to figure out how to separate those different biologies out. And of course, the final point is really converting data into knowledge. Formulating models, they can be descriptive, graphical, or mathematical, depending on the information you have. But the real key is that they're predictive and they explain the emergent properties of these systems. And I'll just give you a vignette of three technologies that are being approached here at ISB, this one in collaboration with Jim Heath at Caltech. And here what we'd like to be able to do is create a microfluidic ELISA assay platform that can analyze thousands of proteins at the atomal level. And to date, we've created one that can do that for 50 proteins in 5 minutes using a fraction of a droplet of blood, and these are actually being used in clinical assays now. But one of the interesting questions this kind of technology raises is if you're to have thousands of ELISA assays, where do you get the protein capture agents, the pairs of protein capture agents that create the ELISA assays? So again, with Jim Heath, we're exploring the possibility that you can take a library of 10 to the 15th D-amino acid 6-mers, that you can use a protein to identify low affinity binders, and that you can use a chemistry called click chemistry to put 2 or 3 or even 4 of those monomers together to form dimers, trimers, or tetramers, and you increase half a log to a log sensitivity and specificity with each mer. We now have trimers that approach the affinity and specificity of the best antibodies. And the real question here is, can we scale this? And a company called Integrative Diagnostics is taking this on now as a major initiative. Can we scale this to actually have this be a replacement for antibodies? The advantages are, one, these are rock stable, and two, once you've used the click chemistry to put together the right monomer combinations, you can use simple copper catalysis to make very large quantities of these trimers or tetramers, whatever you're going to have. And then a final point is another platform that Jim has been developing to use to analyze single cells with regard to 20 different proteins. And he's created a microfluidic platform where you can, with relative ease, analyze at one time 10,000 cells for 20 different proteins. And I'll tell you, this type of data changes the whole dimensionality of how you think about diagnostics, of how you think about responses to drugs and a variety of other fields. So these are the kinds of things that we have to push for the microbiome as well. The analytic tools here, I'll give you just 3 examples. Ilya Stromovich is actually integrating all of the different kinds of genomic data that come out of the Cancer Atlas Project and has done an absolutely marvelous job in beginning to decipher the fundamental essentials there. Nathan Price has actually learned how to integrate metabolic networks with regulatory networks, and that's absolutely a key point in being able to redesign networks and microbes, something we'll return to in a moment. And then finally, Nitin Baliga has developed some very powerful models, predictive models of microbes that he has actually extrapolated to human beings and diagnostics therein, and has some wonderful data on human glioblastoma using the predictive models that have been scaled from microbes. So those are the kinds of things that I think are transformational. So to close this part, let me just run through the 8 challenges that are present, I think, in the microbiome. So one is, how do we do high-throughput, single-cell, global omics analysis? And I would say of genomes, transcriptomes, proteomes, metabolomes, interactomes, and even phenomes. Some of those we can begin to approach now, those on the genomic side. Others are technically far more difficult to approach, and basically many people are adapting microfluidic and even nanotechnology approaches to beginning to attack these problems. Problem number 2 is, how do we not just look at a static picture of an individual cell, but look at the dynamics of the cell in response to signals? So the response can be an environmental signal, or the response can be another cell interacting with it. And again, creating microfluidic platforms where you can look in high numbers at single cells interacting with one another and analyzing globally and subtractively the transcriptomes is going to be a very important thing. We're working with Fluidigm to be able to create a platform where you can take 1,000 single cells, barcode the RNA molecules, and then sequence them in a single run to get 1,000 transcriptomes at one time. So those are the kind of technologies that will be coming online. The omic analyses of communities of cells and those interacting with one another, both in low numbers and in high numbers, and this is going to require new and interesting strategies. You know, one of the really key things in the microbiome is, how do you figure out what all these genes and or proteins do? And certainly one way you can do is a quick one-dimensional homology scan, and that'll give you, if you're lucky, maybe 30% guesses on the genes. But another approach that's been tried that really looks very interesting is to do in silico protein folding. So you do the homology search at the 3-dimensional level rather than the 1-dimensional level. And I think there's some real opportunities there. How do we deal with the signal-to-noise challenges? And that is really going to be a big problem. And the other thing that's a big problem is, how do we integrate different data sets, even of the same type of data, when we know data sets generated in different labs have different lab effects that are far greater than the signal you're looking at? This is something Nathan Price has really been pioneering. How do we convert this data into knowledge? How can we integrate all of this information to create networks of networks, and then ultimately, with sufficient data, transform these into predictive mathematical models? But the final stage is really important, and that's the whole question of, how do we re-engineer the genomes of microbes so we can make them do the chemistries that we're really interested in? And there are really 2 fundamental approaches. So one is what I call the hunt and peck approach, and that's to take 2 or 3 or 4 or 5 genes and plug them into an organism that has some of the right kinds of things, and hope maybe you can set up a synthetic pathway that does something interesting. I think a far more powerful way is to learn the network dynamics of the organism and re-engineer the network so you co-opt the regulatory as well as the structural genes and opportunities and so forth. And of course, how we re-engineer not just individual cells to do what we want to do, but how we then can optimize multiple cells in a community that must interact to carry out complicated chemistries. So I think the idea of generating the information, the idea of generating the predictive models, and the idea of re-engineering microbes is really at the very heart of what we're going to be talking about here today. So with that, let me just close by saying we have sponsors that have generously donated to this symposia, and we thank each and every one of the sponsors that you see listed here. And as well, I'd like to thank all the people that, some of whom put in enormous amounts of time to make this successful, and particularly Dan Rodriguez, who really headed up this whole effort and everything. So I'll close, and then I'll introduce our first keynote speaker for the day, Craig Venter. So Craig is someone I've known for a long time. In fact, I'm not sure when we first met, certainly in the 80s sometime. And Craig and I have shared certain passions together where we disagreed with most of the rest of the world at one time. And Craig, as you know, later went on to show that the ideas we had were right. But to get started, Craig was born in Salt Lake. I think he was, according to his biography, an indifferent student in the early days. He went to junior college and then got his master's and PhD at San Diego. And then he went to Rochester, I think, for a while, and then was at NIH for a number of years before he then set up the J.C. Craig Institutes, and that's now one of the locations is now in San Diego, where Craig actually lives. So it's really quite remarkable if you think about the first that Craig had. So certainly one of the firsts is he really pioneered this idea that you could do partial sequences of cDNAs to get a lot of information about genes at a time when we had relatively little information about genes, and he got very involved in a patent question that didn't really go very far. Number two, he was the first to sequence a whole genome, mycoplasma genome, 500 million base pairs or so, and that was really a pioneering first. And of course, he led the venture on the Drosophila genome as well. He, together with the NIH effort, together did and completed more or less the first cooperative human genome, cooperative meaning multiple individuals were used to get the genomes from Celera, a company that Craig had founded to do this, and the NIH effort. And I have to say, personally, that I was one of the 16 NIH centers, and I decided pretty early on we weren't going to get very big because I so thoroughly disagreed with the philosophy NIH had in sequencing human genomes. In fact, Craig and I wrote one, if not two papers together on this whole thing, and I think Craig really did it right. But he didn't stop there. I mean, he's gone on to begin mining the diversity in the sea and other environmental places. He was the first to actually synthesize the genome of an organism synthetically, and he's using that synthetic genomics now to actually get into the whole question of can we make better and more efficient fuels and everything. So Craig has been called by Time magazine and others among the 10 or 20 or 30 most influential individuals in science, and maybe period, and I would certainly agree with that assessment. So it's a great deal of pleasure to welcome you to ISB for the first time, Craig, and we're really looking forward to your talk.
Thank you very much.